Hello, welcome to Cloudy Nucleus. Here our transcription unit being the section of the DNA molecule to be transcribed into the RNA. On top is the template strand that will be catalyzed to produce the RNA molecule. Below is the coding strand of the complementary nucleotides. DNA exists as a double helix within the nucleus. Now we're gonna make a polynucleotide CAT. CAT. At initiation of transcription, a transcription factor protein attaches to the promoter region of the gene, being the brick fragment. Then RNA polymerase binds, being the stick, to the transcription factor, and starts adding complementary RNA nucleotides to the template strand, going down the transcription unit. This is called elongation. The termination sequence, the new mRNA molecule, is released. Notice how the mRNA molecule is the same as the coding strand, except for uracil being in place of the thymine. In eukaryotes, the pre-mRNA goes through post-transcription modification. This involves adding a modified guanine cap to the 5' end for the quote recognition by the protein synthesis machinery. Poly A tail is added for stability in the cytoplasm. Next happens splicing. Snare proteins with built-in RNA bases bind to the intron and excise not-coding portions called introns, and reglue the exons, the coding portions. They come together by forming spliceosome complexes. Translation initiation. Welcome to sunny cytoplasm. Here a translation apparatus gets assembled. Above is a large subunit, below is small, where the mRNA molecule will thread along. UGC codon for cytosine binds to ACG anticodon on the transfer RNA molecule that brings in the cytosine amino acid attached to its 3' tail. An amino acid is bonded by aminoacyl tRNA synthase enzyme to the tRNA molecule. Amino acid is a subunit of a polypeptide product. As the RNA threads through, being the mRNA, it shifts to the polypeptide side, depositing C to the polypeptide chain, and it discharges the uncharged tRNA at the exit site. A site picks up alanine A. The same procedure follows, adding alanine to the polypeptide chain at the P site. Same with tyrosine. Stop codon here being UAA does not code for amino acid, but triggers the release factor enzyme that releases the polypeptide from the distillation complex. After this protein formation follows, the book vaguely describes it as the polypeptide eventually falls into a 3D shape, so it may carry out its activities. I guess they will learn this more at the university level. Thank you for watching.